Bon, merci. D'abord, un grand merci aux organisateurs pour m'avoir invité. Je suis très content. C'est un plaisir, c'est un honneur aussi. Une occasion pour revoir des vieux collègues ici. I will talk about insect metamorphosis. It's nice because the previous talk by André Nel, which has been excellent, by the way, introduced already the importance of metamorphosis. After the times of Jan Swammerdam in the 17th century already organized the different kinds of insect metamorphosis into three big categories. One of them is ametaboly. Ametaboly means that the insect doesn't change substantially along the life cycle. The example is the silverfishes, for example. This is called ametaboly. Then follows, it was invented, the ancestral of hemimetabolan insects, which were ametabolans, invented hemimetaboly. The insect emerges from the egg with a shape, with a body plan very similar to that of the adult, and during the post-embryogenesis, within the post-embryo period, the insect grows, and only in the last nymphal stage, when it molds into the adult, it acquires the wings and the genitalia. This is the gradual or direct metamorphosis, the simple metamorphosis. And the third category is the most complex, probably the most beautiful, the holometaboly, where the insect ecloses with a body plan which is very divergent with respect to that of the adult. Usually has this worm-like structure or morphology, and it grows, and in the last larval instar, it molds into a pupal stage, which is a kind of bridge between this larval, this worm-like morphology, and the complex adult morphology. The different kinds of metamorphoses are not regularly distributed in the phylogeny. The most basal insects are Ametabolus, Archaeognatha and Zygentoma mainly, among insects, among true insects. Hemimetaboly is distributed in Paleopterans, Polyneopterans, and Condylognatha, or Acercaria. And holometaboly is distributed in the Endopteriota, which are monophyletic. Why is so important metamorphosis, and especially holometaboly, complete metamorphosis? Well, because most of the present extant insects are holometabolan insects. You can see here that more than 80% of present insects are holometabolous. And why such a success? Well, there are different reasons. There is still some controversy about what are the reasons that make the holometabolan insects so successful in evolutionary terms. The most used argument, especially in textbooks, is that the ecological segregation of larvae and adults. They don't compete each other, larvae and adults, and this can be an advantage. Probably holometaboly also helped to shorten the life cycle, which is a great advantage for reproduction, and shorten the life cycles and increase the number of generations. There are other arguments like larvae, probably larvae of holometabolan insects use the resources more efficiently, and possibly the larvae of holometabolan insects can have a better control of developmental processes. In any case, holometaboly or complete metamorphosis was an evolutionary success. You can see here the 4 big groups: Hymenoptera, wasps, bees, ants, 1,000 species. It's really a monster group. Also beetles, almost 400,000 species. Moths and butterflies. And flies, mosquitoes, midgets, with almost 150,000 species. And how metamorphosis is regulated? Well, the first steps that were made to study the regulation of metamorphosis were done by Vincent Wigglesworth, who was evoked by Professor Hoffman in the first talk. Wigglesworth was studying sexual hormones in insects. This was the first goal of this research, using these strange experiments of parabiosis connecting 2 different insects with a capillary tube. So both insects shared the same circulatory system, And he was trying to find that connecting a male and a female, the male could be feminized and vice versa. But this was not true, finally. But he discovered the most important hormones governing metamorphosis: juvenile hormone and molting hormone. You can see here the juvenile hormones, which are very strange. They were elucidated much later, 25 years later, by Reller in the States. He discovered these hormones because he connected a very young nymph with a nymph that was going to molt into an adult, and this nymph was unable to molt into an adult because he realized later that the young nymph was sending repressing factors, humoral factors that repressed metamorphosis. This is the way how Wigglesworth discovered juvenile hormone. Then the other most important hormone is molting hormone. This was elucidated in the 50s, last century, especially by Carl Buchanan and Peter Carlson, that they had to use about 500 kilograms of pupae of Bombyx mori of the same age to isolate some milligrams of a crystal that they were able to elucidate the chemical structure of the molting hormone that resulted to be a steroid structure. We can say that is a ecdysteroid because it's a steroid with a number of hydroxyl groups. It's a steroid, a polyhydroxylated steroid in the end. With these 2 hormones, in mid-1950, 1960, last century, this was the dogma used by all insect endocrinologists to explain insect metamorphosis with these 2 hormones. The juvenile hormone is produced in these prothoracic glands, in these retrobulbs. Well, Ecdysone or molting hormone is produced by prothoracic glands, and prothoracic glands produces the Ecdysone or molting hormone that promotes every molt. And juvenile hormone is produced in these retrocerebral glands called corpora allata. And when juvenile hormone is present with Ecdysone, the molt is in the sense larvae to larvae, is a juvenile molt. And when juvenile hormone disappears, and there is only Ecdysone acting, the insect molts to the adult stage. It's produced the adult morphogenesis. This was the dogma in terms of endocrine terms. But what is behind this endocrine action? Behind this endocrine action is the molecular action of these two hormones. And this was started to be worked in the 70s, last century, 1970, 1975, especially by Carlson, Clever, also Michael Ashburner in the UK. Michael Ashburner especially proposed this model of action of the molting hormone based on the observations of application of Ecdysone and apparition of puffs in the chromosomes of the salivary glands of midges of the order Drosophila, which are giant and easy to observe. According to the apparition and disapparition of these puffs, Michael Ashburner proposed this model. Ecdysone should bind to a putative Ecdysone receptor, and this Ecdysone plus the Ecdysone receptor should induce the expression of early genes, which the early proteins, the proteins from these early genes, would induce the expression of late genes. And this protein, the protein of the genes, would produce the biological effect. In turn, these early proteins would repress their own genes, and the Ecdysone receptor bound to the Ecdysone would repress late genes. This was imagined and proposed only with these observations, but this resulted to be true, as demonstrated later when all these elements were discovered and described and the functions were demonstrated. This was done by a number of scientists from 1975 to 2000, more or less. During this period, the main components of this cascade of transduction of the Ecdysone signal was described. You can see here a summary of some of the most important factors that are induced successively in a hierarchical cascade that starts with Ecdysone bound to Ecdysone receptor. You can see that this has more or less this form of cascade, and these are the interactions among these different transducers that behave according to the Ashburner model that Ashburner predicted. Now the Ashburner model is considered a model which is extended to the genetic action of other factors, especially steroid hormones, but other hormones too. Most of these transducers, Ecdysone receptor E75, HR3, HR4, belong to this very important family of transcription factors called nuclear receptors. Among these genes, there was one which was very, very special. This was broad complex. Broad complex is an early gene induced by Ecdysone, very early, but the function of this gene is different from the others, very peculiar and very interesting. In holometabolan insects, all this was done in Drosophila, mainly, also in Manduca, in Lepidopteran, but mainly in Drosophila. Broad complex is expressed just in the prepupal stage, and this induces the formation of the pupae, a very important factor in the regulation of complete metamorphosis. Without broad complex, the fly becomes inviable in the prepupal stage. The fly cannot go beyond the prepupal stage. And if you overexpress broad complex in the pupae here, when broad complex should disappear, you get adults with pupal morphologies or pupil features. So it's a very elegant demonstration that broad complex has been called the pupil determinant or the pupil identifier. Well, this was the situation, more or less, at the end of 20th century, beginning of 21. We knew that Ecdysone acts through a cascade of transcription factors to produce the biological effect, and we knew most of the components of this cascade. We knew also how is determined the pupil stage, mainly by the expression of broad complex, but we didn't know at all if there was a kind of adult determinant, and we didn't know about how juvenile hormone, the other very important hormone with this terpenoid structure from a chemical point of view, a very strange structure for a hormone, is the signal of juvenile hormone, the anti-metamorphic signal of juvenile hormone is transduced. All this was done mainly in Drosophila, but in the beginning of 2010, 2005, this was a very important technique that allowed many people working on insect metamorphosis to do functional genomics and genetics. All the previous studies were done in Drosophila because you can transform Drosophila, and you can make mutants, you can test, and you can demonstrate the function of a gene, suppressing the expression of this gene and seeing what is the function that disappeared. It's very straightforward. But this was not possible in other insects in general. And this technique, RNAi, interference of RNA, was very useful for many people that were working on other models, not Drosophila, and also working on non-model insects, to do this kind of functional genetics. This is the fundamentals of the action of RNAi, of this mechanism. It's very simple. You must simply inject double-strand RNA, which is identical to a portion of the mRNA that you want to block. And then this double-strand RNA is cleaved into small interferent RNAs. This recognize one of these siRNAs recognize the target mRNA, binds it, and destroys it. So you can suppress or at least deplete a lot the mRNA of the gene that you want to study the function. And the procedure in the lab is really, really simple. With this method, it was possible to see if those discoveries made on Drosophila were also true in other insect species, like, for example, the cockroach. In my lab, we work with this model. Many people doesn't like cockroach. I don't know why, but they are really useful. They are resistant. They respond to the treatments, reliable. So it's an excellent model to do experimental entomology. This is an example of the application of RNAi. We wanted to study if the ECR, the Ecdysone receptor of the cockroach, the homologous of the Drosophila in the cockroach, really had a role in molting, and simply we designed the double-strand RNA, we injected it in the last nymphal instar, N6. We waited until the moment of the imaginal molt, and we checked that the transcript, the mRNA corresponding to the ECR disappeared, or mostly disappeared, and the result, the phenotype was that the insect was unable to molt, as expected. The insect, the control insects injected with a double-stranded RNA, which is unspecific, molted normally to the adult stage, but the animals treated with the double-stranded RNA against ECR didn't molt. They tried it and they did some apolysis. They formed new tissues, but they were not able to ecdyse, to do the ecdysis. In this way, we were able to reconstruct, like in Drosophila, all the cascade of the transcription factor that transduces the signal of ecdysone for molting and eventually for metamorphosis. And the cascade is quite similar to that found in Drosophila, which is not surprising. This is a mechanism conserved from, at least from cockroaches to flies. And the interactions, there are some differences of detail, especially because the cockroaches have 4 or 5 isoforms of E75 with a complex interaction among them, but essentially the cascade and the interactions, the epistatic interactions between them are very, very similar to that found in Drosophila. A very important exception with respect to Drosophila was broad complex, because in hemimetabolan insects, in cockroaches, also in bugs, in hemipterans, broad complex, of course, hemimetabolan insects have no pupae at all. And what is the function of broad complex in an insect which doesn't form a pupae? Well, this should be the ancestral function of broad complex. Broad complex in hemimetabolan insects simply contributes to the formation of the wings, contributes to the growth of wing pads during all postembryonic development, and in the end, if you suppress broad complex by RNAi, what you get is an insect with malformed wings, like this one, with the venation malformed and with some problems in the structure and the shape of the wing. The same, this was previously described in hemipterans by Hilmaz in 2006 in Oncopeltus. This is a 4th nymphaline star. This is the normal 5th nymphaline star. If you suppress broad complex, you get this nymph with the wings that didn't grow, like in control. The same in Pyrrhocorizapterus, another hemipteran. With the advent of RNAi, it was possible to re-study the role of broad complex in holometabolan insects like beetles or even Drosophila. In beetles, it's easy because RNAi works very well in tribolium in this species of beetle. And this was interesting because it was possible to found that broad complex not only promotes the formation of the pupae. Without broad complex, there is no pupae, but broad complex represses the apparition, the formation of adult characters. And this was observed simply studying, examining the phenotypes of this pupae, or this intermediate between larvae and pupae obtained after suppressing broad complex. These animals, they had no features of pupae, or very few, but they had features of adults, which led to the conclusion that broad complex not only forms the pupae, but inhibits the formation of the adult. Well, what about juvenile hormone? What is the signaling pathway of juvenile hormone? This was a mystery until few years ago, especially because this special structure of this hormone, which is a terpenoid, linear sesquiterpenoid, how can be a receptor for that? Nobody knew. Finally, it was discovered that the receptor for juvenile hormone is metopre intolerant. Metopre intolerant belongs to a very different family of transcription factor, BHLH pass. This is a family that was not related at all with juvenile hormone. This was discovered when Tom Wilson were studying the resistance of animals, of insects to juvenile hormone. And finally, it was found that this was demonstrated that MET was the receptor of juvenile hormone, as you can see here. If you suppress MET, the animal proceeds to produce a precocious pupae. If you suppress MET in a larvae, in a young larvae, the insect molts into a precocious pupae. So the receptor, the juvenile hormone, the inhibitory role of juvenile hormone on metamorphosis was suppressed. This was also demonstrated in cockroaches. And this is, well, it was discovered another component of the juvenile hormone receptor, like Ecdysone, which is composed by two components. In the case of juvenile hormone 2, the receptor, the functional receptor, is composed by two transcription factors, MET and Taiman. This was also demonstrated in the German cockroach. And what is beyond juvenile hormone bound to the receptor? Well, the target of this is another transcription factor discovered in this last year, which is Krüppel homolog 1. This is a target of juvenile hormone plus MET plus Taiman. This was demonstrated again with RNAi. If you suppress Krüppel homolog 1, the animal molts precociously into a pupae instead to molt into a larvae. And this was also demonstrated in the case of hemimetabolan insects. Hemimetabolan insects, if you suppress Krüppel homolog 1 in a young nymph, the nymph molts into a precocious adult. This is fantastic. You can obtain this kind of miniature precocious adults. This was demonstrated in cockroaches at the same time and in bugs. This was the state of the art in 2011. Juvenile hormone acts through this receptor. This produces or induces the expression of this factor, which inhibits the formation of the adult. But what is that? What is this black box? This was unveiled in 2014, quite recently, again in the German cockroach, but also in Tribolium and in Drosophila, in hemimetabolan and in holometabolan insects. It was discovered that this transcription factor E93, which is also an early gene of the Ecdysone cascade, that without this factor E93, the animal goes, instead of molting to an adult, molts again to a nymph and again to a nymph and again to a nymph. This shows that E93 promotes the formation of the adult. If you suppress it, the adult is not formed at all. The same year, it was found that if you suppress Krüppel homolog 1, the animal goes to an adult precociously, but if you suppress Krüppel homolog 1, E93 increases a lot. And this means that Krüppel homolog 1 represses E93, which is the adult determinant. So we can see that here is the axis, the general axis that regulates metamorphosis in all insects until now. Juvenile hormone, receptor, Krüppel homolog 1, the main transducer, the master transducer of the anti-metamorphic signal of juvenile hormone that represses E93, which is the master gene promoting adult formation or adult morphogenesis. Yeah, it's the last one. These are the patterns of expression of E93, Brad complex, and Krüppel in hemi and holo, and we can say that this would be more or less the universal pathway that regulates metamorphosis. Well, this is the state of the art now, but there are still many unknowns to unveil how how juvenile hormone drops just in the last nymphal stage. This is a question mark, which is very exciting now. And this is the last one. The students that contributed produce all the work that I described in the case of the German cockroach. Thank you very much.
Thank you very much. So in sake of time, we have time for a very quick question. If there is one. Jules.
Bon, I'm going to ask a question in English to gain time. This was a wonderful story. And I would just, juvenile hormone also is a phytologenic hormone, also plays a role in reproduction. Would this control be the same, or don't you know yet? Control of vitellogenesis.
The control of vitellogenesis is very interesting because vitellogenesis is controlled by juvenile hormone in basal insects, in hemimetabolan like cockroaches and also locusts, but not in holometabolan insects. And in the case of the vitellogenic action of juvenile hormone, the juvenile hormone uses the same receptor, MET and Taiman, but not Krüppel homolog 1. So apparently Krüppel homolog 1 was selected or invented exclusively to transduce the anti-metamorphic signal of juvenile hormone in relation to metamorphosis.
Okay, merci bien.
Pas de quoi.
Thank you.